Now we are going to look at the DNA double helix. So this is a model of the DNA double helix, and we'll look a little deeper. So in 1940, biologists determined that the bases had a specific relationship. So this is a specific relationship between the bases. Adenine base is always connected to thymine, guanine to cytosine. So in uracil for an RNA. So now this is where we are right now. Okay? So the number of purine bases is equal to the number of pyrimidine bases, and this is the combination of those bases. Now we'll see the DNA double helix. So what is the DNA double helix? This is the Watson-Crick model. So we'll look at the model in a little bit, but let us look at some important points here. The double helix consists of two polynucleotide strands winding about each other like a spiral staircase. And I think we can look at that and come back again. So this are two nucleotides. They're winding around like a spiral staircase. So this is the sugar phosphate, sugar phosphate, sugar phosphate skeleton, sugar phosphate skeleton. And these are the bases on the sugar phosphate skeleton. These double helixes are held together by hydrogen bonding between the two bases of the strand that are meeting each other. Again, you can see adenine and thymine always connect through hydrogen bonding, guanine and cytosine. And there are two hydrogen bonds between adenine and thymine, three between guanine and cytosine, adenine, thymine, guanine, cytosine. So they keep repeating. That is the sequence of the bases. So the hydrogen bonds, three between guanine and cytosine and two between adenine and thymine, hold the two strings together. So this is the double helix model. So I'm going to go back a little bit. So the Watson and Crick model said that two polynucleotide strands winding about each other like a spiral staircase, we saw that, contained sugar phosphate backbones. The ribbon or the backbones that we see winding are the sugar phosphate backbones. And the railings, the bases arranged like steps along the inside. The bases were the steps going up the spiral staircase. One strand went from the 5-3 direction, the other second strand went from the 3-5 direction. I think we should see that too. So let us check out our model. We can see 5 ending in a 3, the second string 3 in the opposite, 5 ending in a 3, 5 ending in a 3. So that is the uniqueness about this double strand. Again, I'm going back to one strand that goes from 5-3-, next second strand that goes from 3-5- direction. Now, the amount of adenine was equal to the amount of thymine in DNA. The amount of guanine was equal to the amount of cytosine. Again, the number of purine and pyrimidine bases are equal. We're going backwards in the wrong direction. Okay. DNA contains equal amount of A and T, equal amount of G and C. Adenine is linked to thymine by 2 hydrogen bonds. Guanine is only, adenine only links to thymine 2 hydrogen. Guanine links only to cytosine using 3 hydrogen bonds, and that is very specific. So where are the hydrogen bonds? You can see these are the hydrogen bonds. There are 2 locations here. There are 3 locations here. So we have 2 and 3 hydrogen bonds. A double helix has two strands of nucleotides that wind together, are held in place by two hydrogen bonds that form between the base pairs adenine and thymine, and held in place by three hydrogen bonds that form between base pairs guanine and cytosine. And this is what we were looking at. We can see all that put together, all the information. One string starts from 5 ending at 3. The second starts at 3 ending at 5. And adenine is always connected to thymine using 2 hydrogen bonds. Guanine always to cytosine using 3. And this is the DNA double helix. We see thymine and we know it is DNA. And they are held together by, this is the sugar phosphate skeleton, and these are the base pairs. So this is the DNA double helix. Now, write the complementary study. Adenine always connects to thymine. Guanine always connects to cytosine. So thymine, cytosine, adenine, guanine, guanine, thymine, thymine, adenine, guanine. So let us see. So complementary, they always are connected to the same base. Pairs are always connected. Adenine always to thymine with 2 hydrogen bonds, this 3 hydrogen bonds. So this is your complementary sequence of matching strand in the double helix. So in DNA replication, replication is a cell division. Copies of DNA are produced and that transfer genetic information to the new cells. Remember, one of the very important functions of nucleic acids is storing and transferring of information. So when replication, duplication happens of DNA, which happens during cell division, copies of the DNA are produced and transferred genetic information to the new cells. This happens during cell division. So in DNA replication, the strands in the original or parent DNA molecule separate to allow the synthesis of complementary DNA strands. So we saw the double helix model that is connected through hydrogen bonds is disconnected. So the enzyme helicase catalyzes the unwinding of a part of the double helix by breaking the hydrogen bonds between the complementary bases. So the sequence is now the single strands act as templates for the synthesis of the complementary DNA. So when a part of the double helix is unwound, you have a situation where the single strands now have a template and then create the opposite template, and that is DNA replication. You form a complementary strand of DNA. So there it is, a part of the double helix unwinds, and you are setting up a complementary strand. So this is replication. 2 exact copies of the DNA are produced. This happens during cell division. And now all the information stored in these bases is transferred to the new strand. The DNA polymerase catalyzes the formation of the phosphodiester bond between the nucleotides. The entire double helix of the parent DNA is copied. This process produces 2 new DNA strands identical to each other, exact copies of the DNA. They are called daughter DNA. So now this is complementary base pairing, ensures the correct placement of bases in the DNA, in the new DNA strand. So there is an amazing process of unwinding, replicating, and then formation of phosphodiester bonds between the different nucleotides, because all the different nucleotides have to again connect back into a new strand. So the DNA polymerase catalyzes the formation of phosphodiester bonds between the new nucleotides, the replicated nucleotides, and then the entire double helix is copied, the sequence is copied, and now we have the exact replacement basis in the new DNA strands. Adenine, thymine, how many hydrogen bonds? We said 2. Guanine, cytosine, we said 3. So those are the hydrogen bonds. So DNA fingerprinting, the process, in a process called DNA fingerprinting, what we do is we cut DNA strands into smaller sections. The resulting fragments are separated by size and treated with the isotope, radioactive isotope, that attaches to the specific base sequence. So we get the base sequence and the DNA. All of us have a specific DNA sequence. So that is the DNA fingerprint. If we get the sequence of the bases, we have the fingerprint. One application of the fingerprint is in forensic science, in which DNA samples are connected. So all of us have a specific DNA sequence, and that specific sequence is used in DNA fingerprinting, leading to one use in forensic science. So this is how the fingerprint is set up. This is how it looks. Shows up as a result of the radioactive isotope and the specific sequence, and